In the last chapter, we talked about neurons and their structures and supporting cells. And we started talking a little bit about neurotransmitters and what is starting to happen between the neurons in order to propagate a signal. So now we're going to talk exactly about that. So what happens when an action potential gets to the presynaptic terminal of an axon? How does it cross over to the next neuron or the next cell, whatever that cell happens to be? So the learning objectives for this section are, I'd like you to be able to list the sequence of events at an active chemical synapse in the correct order, starting with an action potential arriving at the presynaptic terminal. And so these are listed in chapter 6 in the book. And that's a good thing. I'm always saying don't memorize stuff, but that's a good thing to memorize. Memorize that sequence of events. It's 7 steps. We want to know what determines how much neurotransmitter is released into the synaptic cleft. And we want to compare the relative speed and longevity of the effect of neuromodulators and neurotransmitters. So we'll start to talk about neurotransmitters and neuromodulators in this section. And then in the next section, we will talk about specific neurotransmitters. We're not going to talk about every neurotransmitter there is. We're just going to talk about the heavy hitters or the ones that come up most often. So we will go into that in the next section. So the synapse is where one neuron meets another or meets another cell. So it's a specialized point of contact between a neuron and another neuron, or a muscle cell, or a gland. Information is transmitted from one neuron to the postsynaptic cell at the synapse. The interesting thing is the neuron doesn't actually touch the other cell. There is space between the two of them. So we have chemicals floating across spaces to make this all happen, which is, it's amazing we can even get out of bed in the morning. This has to happen. So the presynaptic terminal is a projection of the axon that is specialized for the release of neurochemicals. It's formed by the axon and the axon end projection of the cell transmitting the signal. So one single axon on a cell can have lots of presynaptic terminals. The axon can split into different terminals and contact many different cells. The postsynaptic terminal is the membrane region of the receiving cell. So that's where the receptors are going to be that the neurotransmitters will bind. The space between the two terminals is called the synaptic cleft. So all that terminology you should know. Neurotransmitters are chemicals that are produced in the soma of the neuron. They're packaged in small vesicles, and those vesicles go down that axoplasmic transport mechanism to the presynaptic terminal at the end of the axon. And when released, those neurotransmitters are used to transmit information across the synaptic cleft by binding to a receptor in the postsynaptic membrane. So it's pretty cool how that works. There is a electron micrograph in the module that you can look at that has an electron micrograph photograph of synaptic vesicles in the presynaptic terminal. You can see the synaptic cleft and you can see the postsynaptic terminal. So it's a pretty cool picture. You should take a look at it. There are also some videos in the module that are 1 or 2 minutes long talking about what's going on at the synapse, and there's some pretty good animations. So sometimes it's nice, because it's something we can't get our hands on, we can't see, it's nice to look at those animations so you get an idea of what's happening there. So the events at the synapse, the first thing that happens is the action potential comes zooming down the axon and arrives at the presynaptic terminal. So the arrival of the action potential depolarizes that presynaptic terminal membrane, and it opens calcium channels. So we have calcium in the extracellular space. The influx of calcium into the terminal, combined with the liberation of calcium from intracellular stores, triggers the movement of synaptic vesicles towards the release site in the membrane. So then the synaptic vesicles fuse with the membrane and release neurotransmitter into the cleft. So those are the first 4 steps. The action potential arrives, the terminal depolarizes, opening calcium channels. So those are voltage-gated channels, like we talked about in the last section. Calcium floods into the cell, which triggers the synaptic vesicles to move towards the release site, and the synaptic vesicles fuse with the membrane and release neurotransmitter into the cleft. So in the book, there's a little more detail about, and in some of the videos that you'll see, there's a little more detail about the proteins that are involved in the synaptic vesicles binding to the membrane and that sort of thing. But you do not need to know in this detail. This list here is the level of detail that you need to know. So the 4th step, the synaptic vesicles fuse with the membrane and release neurotransmitter into the cleft. The 5th step is the neurotransmitter diffuses across the cleft. So the amount of neurotransmitter released depends on the strength and duration of the stimulus, as opposed to the action potential, which is all or nothing. So with action potential, if you get enough depolarization to reach threshold, you will trigger the propagation of an action potential. However, with neurotransmitter release, if you have a stronger or longer duration stimulus, you will get more neurotransmitter released into the synaptic cleft. So it's a little bit different. The action potential triggers the release, but the more action potentials you have, the more neurotransmitter that is going to be released in the synaptic cleft. So potentially it has more effect. So step 6, the neurotransmitter contacts a receptor on the postsynaptic membrane and binds to that receptor. The receptor changes shape, and the change configuration of the receptor has some different potential. Either it opens an ion channel that's associated with the membrane receptor. It's one of those ligand-gated channels. Or it activates intracellular messengers that are associated with that membrane receptor. So the ion channels that open when a neurotransmitter binds to it, those are those ionotropic ligand-gated channels. That's their job, is just to open it up and let the ions in. The metabotropic, or the other membrane channels, they have farther reaching and longer lasting effects. So you can think of it in this example as either it opens an ion channel or it does something else. And we'll talk about the something else later. So those are the 7 steps I want you to know. So this is the graphic from the book that shows those 7 steps. So step 1, action potential reaches the axon terminal. Step 2, the change in electrical potential causes opening of voltage-gated calcium channels and the influx of calcium ions. Elevated levels of calcium promote the movement of synaptic vesicles to the membrane. The membrane vesicles bind with the membrane and release neurotransmitter into the cleft. Neurotransmitter diffuses across the cleft and binds with and activates a membrane receptor. In this picture, the membrane receptor is a ligand-gated ion channel that opens and allows positively charged ions, probably sodium, to enter the postsynaptic cell, depolarizing it. So the amount of neurotransmitter release depends on the strength and duration of the stimulus, as opposed to the all or nothing action potential. So one of your assignments this week is to summarize the events at the synapse in your own words. So people in the past, students in the past, have come up with analogies. Transportation analogies are popular, or sports analogies. Some people have done videos. I've even had people turn in videos with blooper reels, which were great. So you can use your imagination to come up with anything you can do to describe these events at the synapse without actually using all the terminology. So imagine that you are coming up with a way to describe it to second graders. Second graders are smart these days, though. So, don't dumb it down for the second graders, but in your own words, tell me about these events in the synapse. Most people every year find this to be a really fun assignment, and I'll post one of the videos in the module from a previous student so you can see what they did. But people have done all sorts of things. So there's no limit to your imagination with what you can do with this. So the chemical stimulation of the receptors on the postsynaptic membrane can result in the opening of membrane ion channels. Those are those ligand-gated ion channels. If the synapse is neuromuscular, so we're at the neuromuscular junction, it could also be axosomatic, which means that axon is synapsing with the soma of another neuron. Axodendritic, where the axon is synapsing with the dendrite of another neuron. So if it's neuromuscular, axosomatic, or axodendritic, the flux of ions in the postsynaptic membrane creates a local postsynaptic potential. Okay, so it potentially depolarizes that postsynaptic cell. Or if it's an inhibitory signal, it could hyperpolarize the postsynaptic cell. But in other words, in a neuromuscular, axosomatic, or axodendritic synapse, something happens to the postsynaptic cell. An axon can also synapse with an axon of another neuron, and that produces presynaptic effects. So it might determine whether or not that neuron is going to release neurotransmitter. So it could hyperpolarize the neuron and have an inhibitory effect. So when we talked about gating pain management, this is actually kind of what's happening with gating. We're having one neuron is having an inhibitory effect on another. So depending on where the synapse happens, you can have a different effect and change the potential. But in the axoaxonic is the one that produces the presynaptic effects. All the other ones generate postsynaptic effects. So when we talk about postsynaptic potentials, we are talking about those axodendritic, axosomatic, or neuromuscular, or a neuron that stimulates a gland. There are local changes in ion concentration across the postsynaptic membrane. So at the postsynaptic membrane, changes in membrane potential can either be excitatory or inhibitory to the neuron. So a local depolarization of the postsynaptic cell is an excitatory postsynaptic potential. So a flux of positively charged ions in the cell causes the postsynaptic cell membrane to become depolarized, or less negative than it was before, creating an excitatory postsynaptic potential. So that means the cell is more likely to fire off an action potential. It's an excitatory setup. Local hyperpolarization is an inhibitory postsynaptic potential. An inhibitory postsynaptic potential involves a local flow of either calcium or potassium in response to a neurotransmitter binding to a membrane receptor. So calcium is chloride is generally outside the cell and potassium is inside the cell. So if potassium flows out of the cell becomes more negative because we're losing positive. If chloride, which is negative, flows into the cell, it also becomes more negative. So when it's more negative, it's hyperpolarized, it's inhibitory because it's harder to get that cell to fire at that point. So with excitatory and inhibitory postsynaptic potentials, we're always talking about, are we creating a situation where we're going to potentially have another action potential and cause an excitatory message to go down that cell? Or are we going to inhibit that cell from doing anything? So usually at the neuromuscular junction, and let's just go out on a limb and say always, at the neuromuscular junction, it's always an excitatory postsynaptic potential. At another neuron, it could be excitatory or inhibitory. So at those axoaxonic synapses, they could either be a facilitating potential or a inhibitory potential. So presynaptic facilitation causes more neurotransmitter to be released. Presynaptic facilitation occurs when a presynaptic axon releases a neurotransmitter that slightly depolarizes the axon terminal of the second neuron. So it's going to cause that second neuron to release more neurotransmitter than it would have to begin with without that additional input. Presynaptic inhibition, less neurotransmitter is released. So an axon releases a neurotransmitter that slightly hyperpolarizes the axonal region of the second neuron, and it doesn't release as much neurotransmitter. So if you can get those, it's almost like a double negative sometimes, but if you can get that facilitation and inhibition in your mind, that's a good way to think of it. And postsynaptic versus presynaptic. So when we're talking about postsynaptic potentials, it's either excitatory or inhibitory. When we're talking about presynaptic, it's either facilitation, which is excitatory, releasing more neurotransmitter, or it's inhibitory, releasing less neurotransmitter. So hopefully that makes sense to you. And if it doesn't, then please ask. That would be a lovely thing to ask questions about. We can talk about it in office hours. So neurotransmitters and neuromodulators are all chemicals that convey information among neurons. So a neurotransmitter is released by a presynaptic neuron and acts directly on a postsynaptic ion channel or activates proteins inside the postsynaptic neuron. So it's one neuron talking to another cell, whether it's another neuron or a muscle cell or a gland. Neurotransmitters are used in that one-to-one communication. Neuromodulators are released rather than at a synaptic cleft. They're released into extracellular fluid, and they adjust the activities of many neurons. So you can think of that as the difference between a neurotransmitter and a neuromodulator. So sometimes the same chemicals can act as neurotransmitters or neuromodulators. It just depends on how they're released. So a neuromodulator is circulating in the extracellular fluid, and it's adjusting the activity of many neurons. Neurotransmitters affect the postsynaptic neuron either directly by activating ion channels, those ionotropic channels. That's a typo there. Or indirectly by activating those metabotropic channels and causing activation of proteins inside the postsynaptic neuron. So you can think of neuromodulators as affecting lots of neurons and neurotransmitters as affecting one-to-one. So there are two types of neurotransmitters. The fast-acting, those that are acting directly, and those are those ionotropic ligand-gated receptor channels that open ion channels. They act fast. Transmission requires less than 1/1000th of a second. There are slow-acting ones that act indirectly, and those are the ones that initiate a protein cascade inside the cell, and transmission requires longer. So 1/10th of a second to minutes versus that thousandth of a second. Neuromodulators alter neural function by acting at a distance away from the synaptic cleft. The effects manifest more slowly and usually last longer than those of neurotransmitters, which happen in seconds, and the effects last from minutes to days. So a lot of the medications that have effect on nerve cells, on neurons, act as neuromodulators because they are not functioning at the synapse necessarily. They manifest their effects more slowly, and they last a little bit longer than those of neurotransmitters. So neurotransmitters, their effect happens in seconds, or the neuromodulators, the effect lasts from minutes to days, so much longer lasting effect. So the learning objective, where I said the relative speed and effects of neuromodulators versus neurotransmitters, neurotransmitters act on a single cell from one cell to another across the synaptic cleft, and neuromodulators act on a whole area, on a group of axons away from the synaptic cleft and have longer lasting effects.